Well, thank you very much for inviting me. If somebody can come up and press something here, and then I can move on to the next slide. I think it's a really exciting time.
I can take you back.
Thank you. In genetics, because it's teaching me about Alzheimer's disease, which is what I'm going to talk about.
Yes.
10 years ago, we had some very limited hypotheses made based on beta amyloid deposition in the brain, and that has proved useful, but not the whole story. I think now the genetics, by looking at the full complexity of genetics, certainly I am understanding what the components of Alzheimer's disease are, and they're not what we thought 10 years ago. And it gives us tools to allow us to take those on to try and understand disease mechanisms and predict, actually, those that are at the highest risk. Okay, so this is what I'm going to talk about. So I'll start by going over the susceptibility genes that we've found. And thank you, Mike, because he's laid the platform there, and I don't have to go into too much detail. I'm then going to talk about how we can use that. And prediction, I think, is really exciting, because we now can, using genetics, predict those that are of the highest risk and those that have the lowest risk with over 90% accuracy. That is a significant improvement on our knowledge and provides us with a way of really doing some very exciting work, both in mechanism and identifying people in their 30s, in their 40s, who are likely to go on to develop Alzheimer's disease. And if you're going to prevent the disease starts 10 years before you see any symptoms. That's when you want to get in there with any preventions. So I'm going to talk about prediction. I'm then going to stick my neck out and talk about a new model for Alzheimer's disease. And this is very data-driven. This is what I think the data, the genetics, are telling us about the disease. And it's more complicated, it's multifactorial, it's about combinations of components working together. And that's just something we need to keep in mind. I'm then going to talk about how that can help us understand and identify mechanisms and treatments, and where we are with that in the UK at the moment with the UK Dementia Research Institute. Okay, so when I started out, and Dragana, who's at the back of the room there, wave. Dragana was with me in the early days when we were trying to identify variants that were increasing your risk of developing Alzheimer's disease. This is what we knew when we started some 22 years ago, and we knew that there were mutations in presenilins, in APP gene. There are probably more mutations than I've got on the slide now. There are more being found all the time, but these are very, very rare in the population. We also knew when we started out that there was a very influential risk factor. ApoE is one of the most influential risk factors for any disease, disease with genetic etiology. And that was the sort of starting point. But ApoE is complicated because it has a multitude of functions, and we couldn't, just using that information, nail down what these mechanisms are. But we're improving. In terms of heritability, we know that a common late-onset Alzheimer's disease has heritability of between 58 and 79%. Early onset has a heritability of 90%. It accounts for about 5% of Alzheimer's cases. But this is a very severe, highly heritable form, and it's actually something that we've been working on for the last 4 years to collect samples. We have a sample of 2,000 collected from the UK, and that's going to, I think, tell us a little bit more about some of these rare variants and CNVs that Mike was talking about in regards to schizophrenia. Okay, so I won't go into labour this because Mike has mentioned the sort of design. It is a dead simple design. All we do is we identify variants or SNPs at any particular point in the genome. You have 2 copies of it. You look at the frequency in those cases with Alzheimer's disease, and you compare that to the population frequency, which is indicated here, but those individuals who do not have Alzheimer's disease. And you can see in this particular example, this particular SNP seems to be associated with Alzheimer's disease because the yellow variant is more common in Alzheimer's cases. That's the simplest, that's what we do. But we do that 1 million times for tens of thousands of people all over the world, because we've had to pull together and join with other groups internationally to get the power to identify those variants of very small effect, but very informative about the disease. So what some of our first findings? This was our first paper that had a really significant result. As you can see, you need to have thousands of cases, and you compare those to thousands of controls, and you try and replicate it. You have to have a p-value with about 7 zeros in it to say that you found something. So this is really, really robust, actually, and encompasses a lot of replication. And in some areas where replication is not as robust, I can see, in genetics, that is not the case. So we've got very powerful studies. So we found a number of variants, clustering, PICOM. I'm not going to go into the details because I'm going to try and give you the overview of the picture and what they are telling us about the biology. In the same journal, a French group also found clustering and complement receptor 1 using a genome-wide association study of similar size. This was followed by several others, and I'm not going into detail, but the bigger the studies we were able to put together, the more we collaborated internationally, the more genes we were able to find that were genome-wide significance. And that one had 74,000 individuals. Others found other genes with larger samples. And one of the recent ones that we looked at, and this was a slightly different design. What we did was look at the coding regions of genes and used a chip, and we actually found 3 coding variants. So coding variants are great because we know that those variants are the functional variants that are contributing to Alzheimer's disease. A lot of the variants that we find and that Mike has found in his work occur outside the coding regions, and it's ambiguous. We don't know which of the many different variants in that block of association is actually the functional variant. So we can take these forward, PLCG2, ABI3, and TREM2. PLCG2 is a protective variant, and we've got some really interesting data with that now that shows it has a consistent effect, and that, I think, will go on to form the basis of a target in years to come for treatment. What's come out of that particular study is that a lot of these variants that we're observing are expressed predominantly in microglial cells. Okay, so this is not neurons, but microglial cells. So this is an immune story coming out, and I'll add to that in a moment. The most recent study was published last year, and that also implicated immunity and lipid processing, but also A beta and tau. Now, this is the first time after 5 or 6 years of doing this that we were able to find genes that directly implicated A beta processing. Now, A beta processing was strongly implicated originally by the finding of mutations in the APP genes and the presenilin genes. They're all sort of implicated the processing of A beta, the expulsion of A beta from the brain. So the rare variants of this disease are not exactly the same as the disease we see in the common variants. There are some similarities, but the big factor that's coming out to us is immunity, and I'll come back to that in a moment. Just to finish off, recently there's been a different sort of approach to defining Alzheimer's disease. I'm a little bit wary of it, but it's a very powerful approach. So epidemiological studies like Biobank, I'm sure you're all familiar with it. They took individuals that said they had a parent that had some form of dementia as the diagnosis for dementia. And then they looked at all of those and looked at, did a genome-wide association study with those as the cases compared to the others as controls. This is a very fuzzy diagnosis, and in this context, I'm sure there are people that are wary of that and how you can interpret that. However, some of the SNPs, the variants they came up with, actually coincided with some of the variants that we were finding, but others didn't. So I think we must be a little bit wary about this approach, but it is picking up some aspect of Alzheimer's, but the only way you can interpret it is to add it with our data where you have proper diagnosis of the disease. Mike mentioned exome sequencing. This is where you sequence the coding regions of the genes. That's something we've been doing in international collaborations. We're finding a few genes coming out of that, but it's still early days. It is expensive, and whole genome sort of association studies, whole genome sequencing studies, are getting cheaper, but are still pretty expensive to do. But I think that's something that we will pursue in the future. So taking this all together, this is what we find. So we have a number of genes here. I'm not going to drink that because Mike's been drinking that. So we've got about 40 genes that confer susceptibility. That was the typical Manhattan plot. But what we have now is a lot of rare genes of very small effect, as Mike pointed out. These are those there. We have the mutations that are, if you get one of these mutations, you know you're going to get Alzheimer's disease. But by putting some of these genes together, I just want to point out the one at the top there, which shows it's a polygenic risk score extreme. I'm going to talk about that next, but you can get very close to the predictive value of a mutation if you have a very high polygenic risk for developing the disease. Now, I think that is really significant. So in terms of the GWAS, the data that we have to date accounts for between 30 and 50% of the heritability, depending on the study. So that's a significant chunk of the variance that is explaining and contributing to the disease. Prediction. So I think this is really interesting. What we did, and this is work with Valentina Escott-Price and others, developed a polygenic risk score. This was based on 17,000 cases, 37,000 controls. And what she did was looked at producing an algorithm based on anything that showed any possible associations. So these are effects, p-values of 0.5 and lower. And taken all of those data together, looked for patterns and produced an algorithm with about 87,000 SNPs in it, different variants. And from that, you could predict with reasonable accuracy in the normal populations about just under 80%. You get it right, but that's the area under the curve. But when you actually looked at extremes, your accuracy at the extremes, if you look at the top part of that and the bottom, those with the highest polygenic scores and those with the lowest, you were getting accuracies of 90%. And what is most convincing to me is this particular independent validation was done in samples that were neuropath confirmed. So these are gold standard Alzheimer's cases and controls. And what we see is in that population, we get it right about 80% of the time, but at the extremes, you get it well over 90% of the time. Now, that is really useful information that I think will come into clinical practice. Definitely when we find preventative treatments, but also you may want to consider it now when you're looking at lifestyle choices. We know from epidemiological studies what you do in your midlife in terms of controlling blood pressure, and exercise, etc., may have an influence. So there may be ways that it can be used, but the major advantage is when we actually come up with preventative measures. And we've also validated this in independent samples, the ADNI sample, and it also works there. So what is all the genetics, what is it telling us about what components underlie Alzheimer's disease? So one of the big things I mentioned earlier is inflammation, immunity. That wasn't, up there as a priority for research before the genetics came out. Various people in Southampton, for example, were working on this, but it was peripheral, to be frank. We now have, every time we find new genes for Alzheimer's disease, we find immune-relevant genes. So it's really telling us that Alzheimer has a very strong immune inflammatory component, and I think components. So the complement system is implicated, but there are other aspects of immunity that I think are also going to be playing into this disease. So what we also did was look at pathways. So we looked at what is known about biology. So we took these GO categories, and there are other KEGG categories. These are independently defined biological pathways. And we just married them up with the data, and we looked to see if there were more variants that were associated with Alzheimer's than you would expect by chance in any one of these biological pathways. And this is using the whole gamut of the data set. And yes, immunity is significantly associated. Endocytosis is associated. Cholesterol transport is associated. And other aspects of endocytosis. So we've got components coming out that are giving us cleaner signals, probably cleaner than Mike is getting from schizophrenia, to be frank. This is a study was done with the most recent Concord paper, which was published in 2018. And again, immunity is coming out, but we're also beginning to get some evidence for A-beta and tau. Coming back to the PLCG2 and that point about expression, we are getting many of these genes are expressing themselves in microglia predominantly. So putting this together, this is what I think may be going on in Alzheimer's disease. And it's a combination of these components. I think that's an important thing to take on board, because we tend to look at elements of it. We look at immunity on its own. We look at cholesterol on its own. How does endocytosis fit in? But it's when you get these acting together, and different cell types will have different effects of these different risk factors. So we know microglia are strongly implicated, but obviously neurons die. So we need to know what happens in neurons. Endothelial cells, there's a lot of evidence from epidemiology that your vascular health is important, and that's probably why controlling your vascular health through controlling blood pressure, etc., in midlife may have an influence on this complex set of pathways. Aging, we need to model that, and we need to model immune challenge. We may not see some of these effects unless we challenge the immune system. So there's a lot to do, but now I think we have some very strong directions in which to go to understand Alzheimer's disease. So how can we use the genetics in different ways to help us understand these disease mechanisms? Now, what we usually do is when we find a genetic variant, we can try and model it. And for the APP mutations and for coding variants, that's relatively straightforward. But how do you model the complexity of the combination of these risk factors? Well, I think with iPSC cells now that we can use these pluripotent stem cells, and we can take those from individuals that have perhaps, the high risk, have most of the alleles that are conferring risk for Alzheimer's disease. We can take samples from those individuals and compare them to samples that we may take from individuals that have none of the risk factors. The odds ratio between those two is 150, so we can do biological experiments on those. We will have to do several, probably 20 or 30 iPSC eventually from the different individuals because they have different genetic backgrounds, and we'd have to take that into account. But I think the genetics now allows us to do these complicated experiments where we can look at the combination of these risk factors together. We can put them back into mice, we can look at them in biological systems as well as single cells in a dish. So I think there's lots that can be done in the future. And one of the things that I'm most excited about is, as was mentioned earlier, I'm now directing the UK Dementia Research Institute at Cardiff. There are 7 such centres throughout the UK. And that's what we're aiming to do for Alzheimer's and for Parkinson's and Huntington's disease, to try and understand these disease mechanisms, but based on what the genetics is telling us. Because at the moment, the genetics is pretty robust. I trust it. And we're starting with that information and taking that into understanding disease mechanisms in a variety of ways. And these are the people that are now helping me. And I'm delighted to say we've got 2 immunologists, because we have very few people with immunological expertise now working on dementia. So we've pinched 2 from a different field, and now they're helping us understand what this immunological component of Alzheimer's really is. So we're doing stem cells, Drosophila modelling, a lot of computational biology and functional genomics. That's one of the things that has taught us what the genetics means and how this impinges on biology, imaging, and hopefully maybe gene therapy. And Huntington's, as you know, is very much close to using gene therapy. There are some clinical trials going on, but there's a lot of other possibilities, and that's something that we want to build in for the future. So thank you very much. This is my summary, and I'm happy to take questions. Thank you.
I was just wondering, where does the unfolded protein response fit into the multiplex model of AD?
So what you were talking about is the A beta, yeah, and well, I mean there is a component, but if we just look at the common Alzheimer's genetics, it's a smaller component, but it is a component. So what we need to do is to look at how that fits in with the immune risk that we're looking at, under immune challenge, in various cell types, at various stages of aging. So, once we've done that, I think I'll try and answer your question. But it's not that alone. I think that's been our mistake. It's not so much a mistake. It is that actually we've had very little money to work on this. So you tend to follow, or the field tends to follow one major hypothesis. It's more complicated. We need to look at these together. So I hope I'll be able to answer that in a few years' time. Thank you.
Thank you. Any more questions?
Hello.
Who there?
So as a old-age psychiatrist, I'm seeing someone who's 60 who's got 2 parents who both died from Alzheimer's. How much of this is available to help us now clinically to guide them on risk in terms of genetics?
Well, if you have a strong family history and it's early onset, you can get people can be tested genetically. We know that for a proportion of people, we can say with 90% certainty that they will develop Alzheimer's disease at a time, a later time in their lives. It's for you clinicians to say whether that is useful. It's useful to me if we've got a preventative treatment. Is it useful just to tell people? I mean, there are now with U3andMe, you can get yourself genotyped and you can sort of find out something of this, but not all of it.
That's my question is, so
what can you do?
Not an early Alzheimer's, I'm thinking two people, 80, they both died from Alzheimer's in their late 80s, 60-year-old coming along, worried about his memory, getting a bit anxious about it. And, currently we just say, well, things happen, it's bad luck, and whatever, we'll do the ApoE film or what we do that. If the person says, look, I really want to know what of these are actually available.
Well, there's nothing available at the moment.
Yeah.
But what I'm saying is this is new. This is something that we've only been able to do and validate in the last couple of years. But it's for you to decide where you take that clinically. What I can say is that I can tell you that for a proportion of people, it's not everyone, but a proportion of people, that will have that information. I feel comfortable about that when we have, if we can provide preventative treatments, then that should happen anyway. But at the moment, it's just for their own interest, and I'm not sure it's for your clinicians to see if that is of use to you. The majority of people that you would test, you wouldn't have anything useful to say to them. It's mostly sporadic, and even if you have two parents, it's no certainty, but we can tell them, from this test whether they are in that top few percentages. What do you think?
Oh, I think it's really important in terms of lifestyle. Even though you haven't, like the Americans think, well, if you haven't got something that's going, a monoclonal antibody that's going to prevent it,
yeah.
Well then, why bother? You're just going to worry somebody. But if you've got someone who you know that if you can start predicting life course and time, it then means I can change my lifestyle. I can therefore go on that trip to Australia or South America, etc., retire earlier, do that, plan my life, which I think is really important for people. And some people want to know, and some people don't want to know, and some people like to know. And that's why I was asking, is if there's having an algorithm like that's available for us to be able to say, well, this is the probability, will be very useful for a significant subset of people.
Yeah.
It will also be very important because we're hitting far too late. Like, as you know, adamanumab clears 90% of your amyloid. We've got our anti-tau monoclonal antibodies as well. We're hitting far too late.
Yeah.
We should be hitting back in 40s. By the time it gets to the MCI early Alzheimer's, the horse has already bolted. And so how we can do that, not only with your monoclonal antibodies, but also with your RTMS and your neurostimulation, which I think is the future with it on both sides. But that's why I'm very interested.
I mean, to be honest, that's been the priority in trying to tell pharma that if you're doing clinical trials on preventative treatments, you should be knowing what you've got in your, or actually targeting those at the highest risk, those that before they develop any symptoms, and that's the cohort that I think they'll get most.
You and I are on the same path, and I'd say they don't even do, they're not even doing PETs and things like that, tau PETs and things like that. They're way behind, and I think it's one of the big problems is that, unfortunately, I think the drugs that have got possibility for us are being thrown out because of the bad.
Yeah, I agree.
Bad methodology.
I agree.
I think we've run out of time now, so we will take one more question for
fascinating talk. Neuroinflammation is sort of all the rage now in general psychiatry, as underpinning depression and schizophrenia. So, do you think it's going to be a common pathway there, as the inflammation affecting different parts of the brain or different vessels, or do you think there's going to be a link?
I think inflammation is really complicated. There are going to be many components of it. I mean, I'd be surprised if it's exactly the same throughout. In fact, the genetics for depression the evidence is not there from information from the genetic evidence. But the genetic evidence is fairly weak at the moment. They're not very powerful studies. But I think we need to be looking in this general net. It's about homeostasis. It's really complicated, and immunity can affect a number of the different processes of the brain, and we now need to understand that.
Yes.
Very much. Thank you.
Okay, thank you.